All right, we're going to talk about the plasma membrane. We're going to talk about the structure and the function. Now we are in chapter 3, we're covering the cell. So before we move on, we really need to review the basic structures of the cell. So there's going to be 3 basic structures. When you talk about a cell, the first thing is the plasma membrane. The plasma membrane is always going to be basically the edge of the cell. It's going to separate the internal from the external environment. It's got a lot of different functions. So the first structure is the plasma membrane. Now we are going to be talking about the plasma membrane structure, and we're going to have to zoom in on a section of the plasma membrane to cover it. Now, the second thing is inside of the cell. This whole structure is going to be the nucleus. The nucleus is your command center. This is going to direct all activities of the cell. It's going to have our DNA or our genetic material. And then the third thing, I always like to draw in between these two, between the plasma membrane and the nucleus. Number 3 is called your cytosol. Now the word cyto means cell, and the word sol means solution. So the cytosol is going to have the cytoplasm, which is a jelly-like substance. It's also going to have organelles, and it's going to basically have the cytoskeleton. Now when we talk about the plasma membrane does divide the internal portion of the cell from the external portion of the cell. So when we talk about the plasma membrane specifically, and we talk about the fluid that's in the cytosol, or all of this, is basically going to be filled with intracellular fluid. And the outside is called extracellular, means on the outside, so it's their extracellular fluid. Now, when we talk about the plasma membrane, its function is basically to determine what molecules can enter and exit the cell, which is something that we call selectively permeability. So it determines what can exit and it can determine what enters. That is one of the major functions of the plasma membrane. So, selective permeability. The plasma membrane determines what molecules enter and exit the cell. So, this is one of the major functions of the plasma membrane. But to understand how the selective permeability works, we need to zoom in on the plasma membrane and talk about the plasma membrane structure. Alright, when we say plasma membrane structure, we also say that this is called the fluid mosaic model. Alright, so again, when we talk about the plasma membrane, what we're doing is we're going to basically zoom in. We're going to zoom in on a piece of the plasma membrane, and we're going to draw it out, and that's going to represent the fluid mosaic model. Now, when you talk about the plasma membrane structure, the plasma membrane is made up of 2 primary things. The first thing is it's going to have what we call a phospholipid bilayer. Phospholipid is a molecule. Bilayer means 2 layers. The second thing that it's going to have is it's going to be proteins. Now, before I draw out the fluid mosaic model, I want to go over phospholipids and then the proteins, and then we'll draw it all out together. So the first thing that we're going to talk about is the molecule phospholipids. To understand phospholipids, you need to understand its structure. So when we draw a phospholipid, you always say that it has a phosphate head. Now this is something that you don't usually draw. This right here is its backbone, which we call glycerol, and then coming off of it are 2 fatty acid tails. So it's got 3 structures: a phosphate head, glycerol, which is going to basically be the backbone so that the fat can attach to the phosphate head, and it's going to have 2 fatty acid tails. So this is glycerol. And these are 2 fatty acid tails. Now, the word fat literally translates into the word lipid. So when you look at this structure, it's a phosphate head, glycerol, and 2 fatty acid tails. But when you say fat, that's lipid. So phosphate plus lipid is a phospholipid. So the first thing is we say its structure is going to be a phosphate head, which you can see is this P. The second thing is it's going to have glycerol. Glycerol is literally going to be the backbone. The third thing is 2 fatty acid tails, which are attached to your glycerol. So this is one phospholipid. Now, when we say the fluid mosaic model, we're using this word bilayer. Bilayer means it has 2 layers of phospholipids, which when we draw it, you have to draw one phospholipid and then you have to draw another phospholipid. It's got 2 layers of phospholipids when we talk about plasma membrane structure. Now, the next thing that we talk about are proteins. Now, membrane proteins. There's a lot of different proteins. In fact, proteins have all kinds of different sizes, shapes, anything you can think of, because they have different structures. So the next thing that we're going to talk about, this is my red, is proteins. I have to change markers. This one's dying on me. Before we draw out proteins, the first thing is that we need to understand is that membrane proteins are going to vary in size, they're going to vary in shape, they're even going to vary in function. Now, if you look at the shape of your proteins, that when we draw them out, that's going to help you determine their function or help you remember their functions. So we'll go over that whenever we draw out the phospholipid bilayer. Now, when we talk about proteins, this will be my next thing that I'll talk about. There are two main groups of proteins that we're going to draw when we draw the fluid mosaic model. The first one is what we call peripheral proteins. Peripheral means on the outside, like if you think of your peripheral vision. Peripheral proteins are only loosely anchored to one side of the membrane. All right, the next type is called integral proteins. Now, these are called transmembrane proteins because they're going to span both sides of the lipid bilayer. All right, so we're going to refresh everything we just said. The plasma membrane is called the fluid mosaic model, and it's composed of two things: a phospholipid bilayer. When we use the word bilayer, that literally translates into it's going to have two layers of phospholipids. Now, when we talked about phospholipids, one phospholipid has a phosphate head, a glycerol, and two fatty acid tails. That's one phospholipid. So the fluid mosaic model is going to have two layers of these. And we're going to have proteins that are going to vary in size, shape, and function. So we're going to draw out our fluid mosaic model. First time we draw it, it's going to be simple. The fluid mosaic model represents the plasma membrane. So the first thing that we said is we have a phospholipid bilayer. So you have a phosphate head. Now we don't really draw glycerol, so we just draw the fatty acid tails. So we're going to do one layer of phospholipids. So this is one layer. Now we said it had a bilayer. So you have your phosphate heads to the outside, and then you're going to have phosphate heads to the inside. So you put them upside down to create the phospholipid bilayer. So this is the phospholipid bilayer. Two layers of phospholipids. Now, we also have membrane proteins, which are two main groups. So they're going to be different sizes and shapes. So I'm going to draw a couple different ones. That's one protein. It's a transmembrane or integral protein because it goes to both sides of the phospholipid bilayer. Now, some of your proteins are only on one side. So if it's on one side, that would be a peripheral protein. Putting some more phospholipid bilayers in there. So all of your proteins are going to vary in size and shape. So this one looks like a little door. This one's just on one side, and this one's going to act as a receptor, so something has to be able to bind there. Different function. This one is a peripheral protein because it's only on one side. It's got a different function. You can see that it's got a different shape to all the rest. Integral, 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 peripheral, peripheral. So this is our fluid mosaic model. The plasma membrane, when you're in zoom, is a phospholipid bilayer with two major groups of proteins. Now, I need to cover the functions of membrane proteins. Now, some of them are super easy, and I can describe here with this picture, but some of them are much, much more difficult. So what I've done is I've actually drawn the fluid mosaic model on another piece of paper so that I can do membrane functions. But we're gonna take the notes here and then I'll put this on another picture. So membrane, protein, functions. Now, every time a protein has a different shape, it has a different function. So, the first thing is transport. That one's pretty easy. The second thing is going to be an enzyme. I have notes to put for each one of these. The third functions is signal transduction, which basically acts like a way to signal the inside of the cell. Cell-to-cell recognition. You got to be able to recognize other cells in our body as self or foreign. Attachment of the cytoskeleton, so it's going to help form the walls and anchor all of your organelles. And intracellular joining. So we're going to draw out the phospholipid bilayer or the fluid mosaic model with the different types of proteins, and I'll go over the function. So I've already drawn this out. You should definitely draw it out and then know their functions. So the first thing that I talked about was membrane transport. When proteins form a channel, they're called a transport protein. So when molecules want to move in, the cell has to determine if it's allowed into the cell. Remember, it's a selectively permeable plasma membrane. It regulates what substances come into and out of the cell. So if we have substances, say water or salt, if you have a protein that forms a protein channel, this is going to allow for transport into and out of the cell. So This is providing a channel. So this protein is a transport protein. It forms what we call a protein channel to allow for substances to come into or exit the cell. So that's our very first function. Transport. The next function that we talked about was enzymes. Enzymes are proteins which are going to increase chemical reactions in the cell. So let's just say you have a molecule A and a smaller molecule B, and these need to go through a chemical process. So when you say A plus B yields AB, you use a protein that is an enzyme. An enzyme is a protein built into the membrane that speeds up chemical reactions. So this one right here is an enzyme. Enzymes are proteins. You can see it's an integral protein. These are going to speed up chemical reactions. Now, the way it's going to do that is A and B have to bind to the enzyme in order to make AB. So they'll bind here and here. These are called substrates, and this is the active site. So an enzyme. Okay, the next one is signal transduction. In anatomy, when we say signal transduction, it has a receptor or a binding site that allows for a chemical messenger to attach. This is going to be this one right here. So in anatomy and physiology, we call the substance that binds to the protein a ligand. So this is a ligand. A ligand is a chemical messenger . It binds to a protein receptor . To signal or relay the message to the inside of the cell. Signal transduction binds to a ligand protein receptor. So we had transport, transport. We had enzyme, speeds up chemical reactions. Signal transduction is going to use a ligand to bind to a protein receptor. Now we have cell-to-cell recognition. When we look at the outer portion of the cell, one of your peripheral proteins right here is like a flag. It literally is going to, float in the outside of the cell, and it's going to basically help us know if it's a foreign cell or same cells. Now there's two different types that we talk about when we say cell-to-cell recognition. Here are the two different types. This one right here is called a glyco. It's because it's made out of sugar protein. This one over here is called a glycolipid. Either way, when you talk about the glycolipid versus the glycoprotein, these are used for self-recognition. These are going to act as a flag. So, this is going to be cell-to-cell recognition. This one is also a flag on the outside. Cell-to-cell recognition. So, on this other list, cell-to-cell recognition is going to involve two different proteins. Glycolipid and glycoprotein. Now, if we were using this picture, how would you classify this one? As a glycolipid or a glycoprotein? Well, since this one is not attached to an actual protein, this one right here is a glycolipid. It's a peripheral protein because it's on the one side, but its job is cell-to-cell recognition. So the next one is going to be attachment of the cytoskeleton. So when we do attachment to the cytoskeleton, literally, this guy right here, these peripheral proteins, is literally going to be attaching different microtubules, actin, filaments. So this is the cytoskeleton. So peripheral proteins that are on the inside are literally going to attach microfilaments to help maintain cell shape. The last thing is intracellular joining. When you look at your hand, all of your cells are joined together by another protein. So we basically could draw this out on here. You could draw it right here even and say this is one cell and this is the next cell. So cells are literally attached through proteins, intracellular joining. So over here, we can just literally write a new cell. And the function of this protein right here is intracellular joining. Literally, it just means it joins two cells. So, even though when we say fluid mosaic model, your fluid mosaic model, which represents the plasma membrane, is only made up of two primary things. Now, it does have other things involved, but it's made up of two primary things: your phospholipid bilayer and proteins. Proteins are divided into two types: integral proteins and peripheral proteins. But proteins, even though there's integral and peripheral, they basically have 6 different functions that I've listed. We've got transport. This one right here, we could basically draw the cytoskeleton to help maintain its shape. This one would be signal transduction. This one would be cell-to-cell recognition. This one right here is two enzymes, or it's an enzyme. It's going to take A and B and it's going to join them up, so that would be an enzyme. But basically when you say fluid mosaic model, it's made of phospholipids, integral and peripheral proteins. Now, the shape of the protein determines its function. And to me, it's easier to draw out and be like, oh, okay, that makes sense. That's the fluid mosaic model. Now, there's one last thing that I need to talk about when we talk about the plasma membrane. When we talk about your plasma membrane, maybe two things that I need to talk about. Your plasma membrane is selectively permeable. So when we talk about the function of the plasma membrane, it's going to regulate or allow for transport of substances across the plasma membrane. But before we talk about the types of transports, we do need to talk about the inside of the cell. So the intracellular fluid is the fluid inside of the cell. Intra means inside, cellular fluid. We literally would call this the cytosol. It's on the inside. The outside is the extracellular fluid. It's literally the outside. Sometimes we call this the extracellular matrix. So sometimes we need to move substances from the extracellular space to the intracellular fluid. And sometimes we need to move substances from the intracellular fluid to the extracellular. That's transport. Membrane transport. Now, when we talk about the fluid on the inside and the outside, we need to understand a little bit more about the phospholipid bilayer. So again, this is the plasma membrane. If we did a zoom in on the plasma membrane, if we were to draw this section out, it would look something like this. You would have your phospholipid bilayer with your embedded proteins. Fluid mosaic model. Now, without looking, what's the function of this integral protein? If you've drawn the videos out, you would say it's for transport. What's the function of this one? What's it also sometimes called? It's called a ligand receptor. Ligands are chemicals that bind to a protein to signal the inside of the cell. Now, the reason I'm drawing this out with here is when you talk about this fluid is primarily water. So when we talk about a phospholipid, there's a word that we use to discuss a phospholipid. A phospholipid is what we call amphipathic. An amphipathic molecule is a molecule has one region that is hydrophilic. Hydrophilic means water loving. And one region that is hydrophobic, water-fearing. Now, because you have primarily water on the inside versus the outside, when we look at this, this would be extracellular fluid, which is primarily water. And on the inside, this is intracellular fluid, primarily water as well. So, when we talk about a phospholipid is what we call an amphipathic molecule. The heads The phosphate heads love water. So those are hydrophilic. They love water. Hydrophilic literally means water loving. Now, hydrophobic means water fearing. So all these fat, if you think about fat is hydrophobic. So these face in towards the inside. The fatty acid tails are water fearing. So fatty acids are lipids. And when we think about fat, fat does not like water, so these are going to be hydrophobic. Now that's important when we talk about the cell and what can pass through certain areas of the cell. So that is all I'm going to cover on plasma membrane structure. I'm going to make another video about transport because we know that the cell is selectively permeable. Meaning it regulates or allows some substances to enter and some substances to exit. However, when you talk about the plasma membrane and allowing stuff to be transported, that's called membrane transport. So there's different types of membrane transport, but before you can learn the types of transport, you really need to understand the fluid mosaic model. Not just the different types of proteins, but understanding its properties of being amphipathic with hydrophilic phosphate heads, which face in towards the intracellular fluid and face out towards the extracellular fluid, but everything that comes with it before you do transport.